too heavy. I just wanted to ask you from last time, just to start it off with, because you in regards to universal common descent, you said that obviously like a population, say, of dogs today, and obviously I understand the law of monophyly. We can never outgrow our ancestry, but a population of dogs today would have ultimately come from, say, a population of non-dogs, right?
Yeah.
My question is, because in understanding the law of monophyly, like obviously like a eukaryote will never stop being a eukaryote, a mammal will never stop being a mammal, and obviously on and on and on. But would, given enough time and enough change, I guess enough evolutionary processes, could a dog today eventually become something that's, say, recognizably a non-dog to somebody?
I don't know what you mean by recognizably a non-dog. If what you're asking is, could the descendants of current dogs ever become something that, if you were to just see a picture of one randomly, you wouldn't immediately say, "Oh, that's a dog"? Then I suppose yes, but it would still be a dog and recognizable as such by a taxonomist.
Okay, so recognizable by a taxonomist is obviously a dog, but say the non-dog, that dogs today came from, say long ago and far away, like would those be recognizable to a taxonomist as a dog that we would recognize today as a dog, or no?
No, they would be recognizable as a more basal form.
Okay, so if we're looking at, say, a population of dogs
And I
today,
just to clarify that point, dogs today are recognizable as within the same group as that more basal form. In other words, the dogs today have derived features not seen in the more basal group, but they have all of the features that are seen in the basal group.
okay,
Right, possibly with some increased specificity or derivation, right? But there's no you don't see highly derivative features in the progenitor population that are completely absent in the later populations, generally speaking.
Okay, no, and good answer. I understand what you're saying. So I guess it would come down to, like, is there any, and I think I asked you this before, like, is there any limits then to obviously the change that we could see, say, in just a population of dogs? I'm just using that as a generic example. Obviously, from your answer before, you indicated that yes, there's probably, a physical, based limit. But
I would say that there are soft limits arising from pre-existing regulatory schemes, right? So that there might be certain roadblocks to certain varieties of change that would be very hard to overcome. For instance, having an extra pair of limbs would be a real trick, right? It's not.
yeah.
It's not that it's strictly speaking impossible, but getting there would be extremely difficult. I can't right now sitting here come up with a reasonable pathway to get an extra pair of limbs, right? I'm not saying it's impossible. I just don't know how it would work.
Okay, no, and I understand that. If we look at, say, I know I gave the example of a mouse before, obviously because it's a mammal, dog's a mammal, and elephant's a mammal too. Like, do you think, given enough change, evolutionary processes, environmental pressures, like, do you think we could ever get, say, a dog as big as an elephant? Because obviously we know there's animals as big as an elephant, like an elephant. So do you think that's possible, or would there be a limit there as well?
Yeah, generally speaking, I think that changes in size, so long as you obey certain things for health reasons, that would be possible. A lot of the problems when you look at the really large breed dogs is that they are bred to be big but still proportionate as dogs, right? So,
Yeah.
so the bones are bigger, but they're still the same proportion as a normally sized dog. But that's actually not big enough. They need to be proportionately much thicker to be big enough to support the weight. And that's a big part of the reason why big dogs have so many problems with their bones is because their bones just aren't big enough to support the weight, even though they look proportionately like they are big enough. And if you ever see like bones from an elephant, they're, you think an elephant's a really big animal, but like the femur from an elephant is much bigger than you would think it would be, because it has to be. Because of the cube square law, right? That the strength of the bone increases with the cross section, which is the square of the size, but the actual weight increases with the cube. So the weight that this has to support increases much faster than the strength if you're just growing proportionately. So
okay, and I understand. So I guess my follow-up question, and I guess my follow-up question would be, given, say, the biblical base model, like obviously we, would predict, clear limits and a clear barrier, obviously, to the change we see, whether it's physical, whether it's genomic. And we seem to be in agreement that, there are obviously some limits. Like, would you say billions of years ago, obviously after the fact of, abiogenesis, whenever we got that first, living cell, that first single-celled organism, would you say that the planet was ruled by, whether it's bacteria, single-celled organisms? Would that be a appropriate inference?
I don't know what it would mean to say that the planet was ruled by single-celled organisms. That seems like a poor use of words.
Well, as in, like, was there only single-celled organisms? Whatever that single-celled organism was would have been our, common ancestor, whether there was multiple or a single, to all life we see today, right?
That seems right, yeah.
Okay, so my question would be, like, based on that fact, billions of years ago, say, the Earth was ruled by, these single-celled organisms, bacteria, whatever, but now, billions of years later, we've got, such a wide diversity of living creatures, organisms. Like, wouldn't, according to the model of universal common ancestry, wouldn't that predict, no limits? Because if you're going from, say, a bacteria up into, a whale, obviously indirectly, many, different steps and changes, how can there be limits to the change? Go ahead.
Well, again, I'm saying that there are soft limits, and that these limitations are based on pre-existing pathways. In other words, when you talk about what's possible, you're always really talking about what is possible within this particular context. So if you take the example of the dog, right? So the dog can't just get bigger, right? Because the dog has to have food. The dog has to survive within its current environment, and it has to do so in more or less the same way that it has been surviving, especially if you're removing human influence from it, right? So when you're talking about this, it can't just, it's not as simple as saying that you just have a selective pressure for size, because why would you just have a selective pressure for size? There must be other environmental concerns at the same time, and those things are going to be limiting factors. Now, at the same time, there are circumstances under which, in total, the selective pressure would be towards a greater size. For instance, if the average size of the prey was larger and they weren't going to be working together as much for whatever reason, right? So, obviously, there's a common ancestor between dogs and bears, and this common ancestor was a carnivore, right? It was hunting things. Why did one group stay? Dogs are bigger than if I'm remembering right, than the common ancestor of dogs and bears, but they're not too much bigger. But bears are much larger. So what happened there Well, because bears were doing something very different than what dogs were.
Okay, so I think I'd have a few things to say. So what I'll ask three questions then. So one, wouldn't a lot of what you're saying kind of be more of of a story? So obviously, if we're not observing a lot of these things and a lot of these possible situations that could lead to these types of changes, then it kind of just remains in the realm of story time. But secondly, wouldn't like a lot of the breeds that we see, say with the dogs, like I think there's what 360, maybe 360 to 400 recognizable breeds of dogs. Obviously, that's based on artificial selection. So as humans, as breeders, we're selecting certain traits and for bigness, smallness, obviously, long hair, thick hair, whatever it is there. So I mean, obviously, if we're bringing about these changes versus say nature bringing it about, how what are the chances of nature itself bringing about such extensive changes? And lastly, what about like a study? I'm not sure if you've seen it, but it came out recently. It was a genetic study that seemed to indicate and reveal that 90% of species arose recently. So that would seem to suggest, or at least reveal, genetic boundaries. And even in regards to the modern dogs, like the breeds I was talking about, most modern dogs did come from only a handful of breeds, say in the 1600s. And that's a lot of change, in only obviously a few hundred years. Go ahead.
Yeah, so in no particular order. Yeah, so if you're doing artificial selection, you can work very, very quickly. That's should be pretty obvious why that's true. I mean, I don't think that I really need to go into detail on that. Yeah, you can get a lot of variety very quickly if you're doing selective breeding, especially when you have, pre-existing variety within the different groups of working dogs. That there had been artificial selection of a kind for millennia prior to that. Because we were developing different lines of working dogs, but they were doing different particular jobs, and so they were selected for different purposes through this process. And so if you start then crossing those that have been bred out for different purposes, and then you start picking out very particular traits, then yeah, you can move very, very fast. But ultimately, the amount of difference between any of these things is actually very small. People like to think that all the different breeds of dog are so very, very different, but actually, if you look at the skeletons, there's not much difference. The skulls tend to vary quite a bit, but the rest of it is really just general scaling, and that's not the most impressive. That's not really a good marker of variety. To the other question, isn't it a story? Well, the thing of it is that usually I'm faced with a question of that I'm being told that evolution or whatever is actually impossible, right?
Sure. Yeah.
And
Actually, before you go on, can you just give me like your depth? Because I think the word evolution is a bit of a slippery term. Like my definition of evolution would be like a change in allele frequency or gene frequency in populations over generations, right? So I agree that we're seeing change.
The whole thing, right? All of it, common descent, every last bit of it, right?
Okay, so the whole.
Yeah. So if you're going to say that type of change itself is impossible, right, then I don't actually have an obligation to demonstrate that any particular mechanism actually happened. All I have to do is prove that there is a plausible pathway. Because if there's a plausible pathway, you can't say that it is actually impossible anymore.
Okay, so would you admit that a lot of what is said, say even a lot of which you said earlier, is more based on a story? Like obviously it's not something
Well,
that's
it's a
written in
story that
stone.
demonstrates possibility, right?
Okay, so it's a plausible story based on what, you
Right.
know, we
Because
know
the
today.
thing is that I am not like my specialty is not taxonomy, right? It's not phylogeny. It's not paleontology. It's not any of these other fields. I do not have in the back of my mind a deep catalog of different organisms that have existed and when they existed and where they existed relative to one another and how they differed from one another in minute detail. So there are people who can give lists of organisms and show that, you know, here's this gradual gradation of change, and yes, they lived in overlapping areas in succession. Um, I can't do that, but what I can do is give a plausible pathway. And if there exists a plausible pathway for the thing, you can't say it's impossible,
Okay.
which is the claim, the specific claim that I'm responding to. I'm responding to the idea that the thing is impossible in entirety, which I don't agree with.
Okay, no,
Um,
and I understand what you're saying. I think I would say back to obviously the definition of evolution. We seem to agree on that. You know, obviously then you're expanding it into obviously universal common ancestry. You know, the fact that whether it's banana plants, humans, monkeys, and elephants are all related through common ancestry. But I think it all comes down to limits. So obviously based on that study that I'm not sure if you're familiar with that study that
yeah,
did.
actually, I am. I was gonna get to that last.
Okay, so I want to ask, if it does come down to limits and comes down to a clear barrier and boundary, and obviously I want you to comment on the study that seemed to reveal that 90% of species arose recently and obviously at the same time. Also, would it be empirical science to say that, although, we see dogs produce dogs, bears produce bears, pine trees produce pine trees, is it scientific to say that bears, elephants, dogs, and pine trees are related through common ancestry? If a lot of it is, say, based on story, then, and I get, you're building a plausible story,
Well, no,
but it
so
doesn't
it's not.
necessarily mean the story is true. Go ahead.
So yeah, okay. I got to respond to that first before I get into this thing about the when different species arose. Um, it isn't that it's based on stories. It's the actual evidence is based on the data of the fossil record and the genetic record and everything else. That's not a story. That is empirical fact. Okay. I don't have that information committed to memory, so I can't rattle it off for you on command. So what I can do, if you're talking to me, right? What I can do, I'm limited to the information that I have at my disposal, and also what I can work with in terms of demonstrating plausibility. But that doesn't mean that that's all there is. It just means that's what I've got, at hand.
Well, I mean, if I had if I have a question, even if you had that information, wouldn't it take like a long time to lay it all out?
Oh yeah, of course, and that's another issue as well. Is that yeah? Whenever you're talking about this, people always want to go. Whenever I hear people say that they want to, we just want to go through evolution really fast. It's like saying that we want to take a walking tour of the continental United States. Like you just can't do it, right? You can't do the entire topic in a short period of time. You've got to go. It just takes so much detail to really lay all of it out. I
I would
mean,
say, yeah, I would say it would be fair to say it would they want to take a walking tour of the entire planet, but
Okay,
that
well,
might be.
let me just respond real quick then. I think, and I understand, you would look into, paleontology, the fossil record, the geologic record. Obviously, you're going to look into a lot of other fields to have that, extra evidence to obviously go with the theory. But the thing is, if we're looking at, say, a genetic level, and we do see that there's, clear limits, a clear boundary. We see that, and I know you don't agree with genetic entropy. We'll get into that. Let's say we do see this accumulation of these near neutral deleterious mutations that are degrading and obviously degenerating our genome. And obviously the fact that it wouldn't be easy for, say, a simple single-celled ancestor like we were talking about earlier to, say, evolve into a human over billions of years, because obviously novel traits would have to be gained, right? New anatomical structures like brains, arms, say legs, plus new functions. Like, for example, cardiovascular and muscle activities would have to be acquired. So it seems like there's a lot of things genetically that would
wrong stuff I got to respond to, or what?
So what I'm saying is, if we see on a genetic level, based on everything that I said, that obviously molecules to man evolution can't be true,
we don't see that.
That means going to the fossil record, going to paleontology. That's kind of circular, because now you have to assume that ponds come to people evolution is true when it's really not true. Go ahead.
Right. So we don't actually see any of that, though. That's the big problem. Is that all of that stuff you just said, every last bit of it, is actually not in the evidence, right? We, firstly, you were talking about, and it's just I laugh because it's funny. You said develop a new leg, right? That's like, what's novel about a leg?
Well, a new anatomical structure,
right?
Like
okay, so let's look at a leg. Let's look at a leg. If you have the fin of a sarcopterygian fish, okay, and you have a basal tetrapod limb, name the new bit of anatomical structure to get from A to B.
Well, for one, going from A to B has never been observed, so now you're going to resort to
that's not what you claimed. You claimed there was a new bit of anatomical structure in a leg that's not found in a fin of a sarcopterygian fish.
No. If you go back and rewind, I said for a simple single-celled ancestor to evolve into a human over billions of years, novel traits would have to be gained, such as
a leg.
I never, such as a leg. So obviously, if the Earth, which you admitted was ruled by single-cell organisms, but now we have
hold on. Bacteria don't develop a leg.
I understand that indirectly your bacteria went to a whale, but it's still unscientific.
So what we're talking about is, what is the novel anatomical structure required to get from a sarcopterygian fin to a basal tetrapod limb, a basal tetrapod leg? Is there any novel anatomical stuff there? And if there's not, we can move it back a step and talk about whether or not there's any novel anatomical structures required to get from the to get to the sarcopterygian fin from the prior structure there. And you'll say there's not anything novel about that. And we'll move it back a step further, and you'll say there's not anything novel about that. And we'll keep moving back until we get to sponges, and you'll say there's not anything novel through all of that. And then you'll say that there's not anything anatomically novel between a sponge and a bunch of vaguely colonal what's the word?
Okay, so could you
flagellates. So basically at no stage from single cell all the way to human is there ever a single little bit of A to B novelty in terms of anatomy. There's novelty in terms of anatomy if you go over a huge stretch, right? But from A to B, there's no novelty ever.
Now, can you point, say, to the DNA sequence where this new trait, so you're not saying it's a novelty, but say this new trait sequence is found? Because obviously if we looked at DNA these days compared to, say, the fossil record and, dead things long gone far away, can you see the DNA sequence?
Several things.
Go ahead.
Several things. First, generally speaking, we don't expect any reasonably complex trait to be driven by a single gene. Second, moving the goalpost, right? Because that's not what we were talking about. So
Well, because you're trying to say you're going to go from this fin to obviously, you know, more of a tetrapod-like
No, you said that
leg.
you said that you would have to have anatomical novelty, but there is no anatomical novelty in it. So it's not true that you have to have anatomical novelty.
Well, for one, we don't see these types of changes today. So you're
But
saying
that's
these
not
build
that
up.
is not the claim. That is not the claim I was responding to.
But are you saying that based on these
And actually, we do
accumulation
see
of
those
changes and variations, that's how we're going to go from, say,
Well,
fish,
we
amphibian,
do
reptile,
see we
mammal? Is
do
that
see
how it's going to be slow, right?
so we do see exactly those types of changes. For instance, there's a known mutation that I've referenced before to Tommy. I would have to look it up now again, but I can tell you exactly what it does. It very slightly increases the length of the long bones, right? So your femur and your humerus will be slightly longer if you have this mutation. So the point of that is that it's changing relative proportion of anatomy, because that's all that anything ever needs. That is the only thing that ever needs to happen. Okay? Because if you can change the relative size and proportion of bits of anatomy, you can get a tetrapod limb from a Sarcopterygian fin, because all the bones are already there in the same relative positions, developing in the same relative order, under the control of the same regulatory elements, which is the third bit that I was going to respond to. Because yeah, actually we do know a lot of the regulatory sequences that are responsible for this, because if you knock them out in mice, you get something that is more similar to a Sarcopterygian fin than a proper limb. Yeah,
Now,
we actually do have it.
but what I'm saying is, and I agree that not one gene is obviously going to mean a leg or a fin, but I'm asking, and I don't think it's, you know, changing the goalpost. I'm just asking for sequence of genes which would actually point to this, you know, change in, you know, a new leg or new fin. But because I'm just saying, if that change is that big, it should be noticeable in the genome. Because correct me if I'm wrong, that is worth millions of DNA letters. So
You are wrong.
obviously it should be noticeable. But how come we don't see that? Go ahead.
Well, because I mean, you actually are wrong. So this is one of the really common misconceptions about things, which is that these regulatory systems, right, don't do much at first, but they're built off of. So you have a regulatory system that initially only causes a small change, but then other things latch onto it. And then once you have all of these other things that are built off of it, if you remove that one thing that initially had only been doing some minor thing, it messes with all of the stuff that came after. It's like imagine if you're building a house of cards, right?
Oh, it seems like I'm doing a lot of imagining with the storytelling here.
This is an analogy. So
Okay,
if
go ahead.
you have a house of cards, right, and you build like a small wing of this house of cards, and then you decide you don't like it, and you just remove it, that's fine. It doesn't mess with anything else. But if you do that same thing, you build that little wing, and then you build a second and a third and a fourth story, and then you decide, no, I don't like that wing, I'm going to remove it. What happens to the stories you built above it? What happens to the rest of the building? It's all going to go, because it wasn't vital when it first developed, but you build off of it, and those things stacked up on one another become important. And actually,
Okay, so-
yeah, you don't have to imagine that, because if you knock this out in more basal forms that currently exist, it doesn't do that much damage. It has a very minor effect. But if you knock it out in mice...
I just want to clarify. So you're saying that obviously this stacking is analogous to, say, these, you know, minor accumulation of changes and variations that are leading to these.
Not specifically, no. No, what I'm talking about is that you have successive layers of regulatory control, right?
But at the same time, you have successive changes that are going to take your non-dog population into an actual dog population.
Let me put it this
it's
way.
all successively built on changes, right? That's what you're saying. One
It
information
is, but
adding mutation at a time.
it is, but what matters is the type of mutation that we're talking about here. So you can have a set of transcription factors, right? That at no point does the sequence of the transcription factors themselves ever change, okay? But you have the binding motif for one transcription factor, right? That that occurs in association with a particular gene, right? And so now that particular gene is being expressed under the regulation of that transcription factor, factor A, let's call it. Okay, now if that gene that is now under control of transcription factor A is modulating transcription factor B.
Is this something that you're observing in the lab, or is this
we can see
something
things
that
like
we're imagining? I just want to
type of
clarify.
regulatory system in real time, in real life? Yes. And so it's not that you have the successive development of new genes per se, but you have a the addition of a layer of regulatory control through changes in regulatory regions that then affect and or are affected by later layers of regulatory control, and then still later layers of regulatory control.
Okay, so you're saying this is all observable currently? Like you can look at this in the lab and on a genomic level?
Sure. Yeah, this is I don't think that any of this is terribly controversial.
Well, what I'm saying is this going to lead to, and I understand you know, it's obviously small and you know, going from a fish fin to a tetrapod leg is not, you know, a novelty.
I'm glad you agree.
But what I'm saying is, if we look at these mutations, okay, I believe they come in three forms, right? Neutral, detrimental, or obviously beneficial, whether it's beneficial to a given environment. But how many, what's the percentage of mutations that are actually deleterious, that are going to affect the organism in a negative way, versus one that will help this animal proceed, you know, with what you're saying, with new traits and new novelty?
I can't imagine how you would ever even begin to come up with estimates to those numbers. And
would you say the majority are detrimental, though? Like, for example, do you believe that there's a truly neutral? Would you say there's a truly neutral mutation? Do you believe those exist, or do you believe that every mutation?
Here's the real problem. Here's the real problem with the description of this, which is that for any approximately neutral mutation, let's say that there are no truly neutral mutations, just for the sake of this argument. Okay. Sure. So for any approximately neutral mutation, the effect, if it's approximately neutral, must be very small. Whatever effect it has, it must be very minor.
Yeah, I agree.
The thing about very minor changes is that whether they're beneficial or harmful is entirely dependent on context. If you're slightly bigger, that could be good or it could be bad. It depends.
So it's like environmentally dependent, right?
Yeah, it's very environmentally dependent. And so if you want to say that there are no truly neutral mutations, which I would definitely disagree with, but I'll just give it to you ad arguendo. Then,
well, I don't want you to just give it to me. Let me just ask you, because I'm thinking, you know, by its very existence, a nucleotide cannot be neutral. So I don't want you to just agree with me. And why I believe that is because nucleotides, they do take up space. They affect spacing between other nucleotide sites, and they even affect regional nucleotide composition, you know, what else? DNA folding, nucleosome building. So that means even if a nucleotide contains absolutely no information, it still cannot be neutral, because it's still going to slow replication and even waste energy. So I'm just saying it seems evident that there are really no neutral mutations. Like, would you agree with that, or would you still have to concede that?
Yeah, so you're confusing the fact that there are regions where spacing is relevant for the and you're taking that and you're running that and extrapolating it out to assume that all spacing is relevant. and that is demonstrably false.
Well, I think it also goes back to obviously the non-coding DNA, because you guys would say that the non-coding DNA absorbs the mutations, right, making them neutral. I know we talked about that
Oh, and also
extensively before, but I want you to comment that. But what I'm saying is, if this non-coding DNA, let's just say it does help during embryonic development, and it can be part of many diseases, which obviously they're finding out now. And just because scientists cannot see any function per se in some of these non-coding DNA regions, that doesn't mean they're non-functional. So that would make the majority of mutations near neutral versus, say, neutral, right? Go ahead.
Right. So what you just said is that if this is true, then something else is true. And the problem is that not only is there not evidence that it is true, there is strong evidence that it's actually false.
Well, if we look at, and I want to give an accurate representation of what ENCODE found, and tell me if I'm representing it right, okay? So ENCODE would have estimated that 80% of the genome is functional, but in the sense, okay, and I get what you're saying, that it has specific biochemical activity, right? So I think it was an ENCODE researcher that said, it's like opening a wiring closet and seeing a hairball of wires. We try to unravel this hairball and make it interpretable. So he's saying that obviously a lot of this function, it's believed to be regulatory, right? So the DNA is telling the genes when to produce the product.
No, that's not right. I'm sorry, that's not right.
Okay, go ahead.
It's not believed to be necessarily regulatory. When they say that it has firstly, there are two errors there, which is it isn't that the single nucleotide is involved in specific biochemical activity necessarily. Instead, what they're saying is that these nucleotides are within some distance of something that is involved in a specific biochemical activity. That's the first distinction, right?
And would that fall back to your spurious transcription?
No, this isn't. We're not even there yet, right? So basically, what they're saying is, let's say we know that some transcription factor has a certain consensus motif, right? So I'll just make one up. There's a transcription factor that binds to, and I'm going to type it in the side so I don't forget it. It binds to GCA TGA. That is the consensus sequence for this transcription factor. So what ENCODE would say is that every time you find GCA TGA in the genome, you go 1 kilobase in front of it and through 1 kilobase behind it, and you say that 2 kilobase region, all of it is involved in a specific biochemical activity. Now, the fact that there's a binding motif doesn't mean that you ever get transcription from there. It just means that it matches the consensus region of a binding motif, right?
But how come we find all independent functions, the ones we're looking at, they seem to be in that 80% area?
Well, because the 80% area includes everything almost, except for the only thing that it doesn't include is that other 20%.
Okay,
right? So what are the odds that most of the stuff is included in 80%? Well, it's pretty good.
But if every month we're seeing more, okay, for one, I want to go back. So obviously we agree that ENCODE did define function as specific biochemical activity, right? I'm not
that is what they defined it as, but I don't think that we have sufficiently explained what they mean for the context of this conversation.
But did ENCODE find the majority of DNA is transcribed into RNA? Copying the DNA into RNA? Because it was thought to be exclusive to parts of the DNA that coded for proteins.
That's false.
Now we're seeing that activity in the non-protein coding region.
No, I'm sorry. Spurious. Now we are to spurious transcription.
Yeah, so now it goes back to
transcription was a known phenomenon for decades before ENCODE. Now you could say that they didn't know the entire extent of it, but they knew about it.
But what I'm saying is, would you be disagreeing then with a lot of the ENCODE researchers? Because I know one said, based on the findings, he said, we are the most complex things we know about. It's not surprising that the manual is huge. I think it's going to take this century to fill in all the details. So what he's implying is that it would take a century to understand 100% of the DNA language. So it seems like a lot of the quotes and writings of these ENCODE researchers, it seems like they're doing their science, without that evolutionary bias, and they seem to be suggesting that our genome is chock full of functional information. Like, would you disagree with that quote, or do you think that that's just a misrepresentation of what he's
You're
saying?
asking me to speak on a quote from someone I don't know in a context that I'm not familiar with, right?
Okay, well,
like,
okay, if you're saying spurious transcription then, but we're seeing that much of the 80% say was found to be controlling also the expression levels.
that isn't what was found.
No,
I'm sorry, you're simply wrong.
we know but we do know that non-protein coding DNA is telling the protein coding genes when to produce their product, aka the protein, and when not to. So
again,
that is a clear trajectory.
Here's a fundamental error in your thinking, right? Which is that just because some things within category A do thing X,
okay,
it
but
doesn't mean that all things in category A do thing X.
But it seems like you never agree with the specific expressive and regulatory roles in these specific functions I'm describing. You do agree that those are found in those non-protein coding regions, though, right?
I'll give you an example. There is a gene, XIST, I believe, is the symbol for it, which is involved in sex differentiation, if I'm remembering all of this correctly. And that is a long non-coding RNA. Is very important to proper development, but it's not a protein. Okay, now the fact that that exists doesn't mean that all long non-coding RNAs have very important functions. It means that it is possible for a long non-coding RNA to be functional. Doesn't mean that just because you found a long non-coding RNA that thing does actually have a function. Now, the same applies to all of the other examples that you might point out. The fact that it is possible for something of a certain description to have a function doesn't mean that all things of that description have a function.
Okay, so when you look at things like, regulating DNA replication, regulating transcription, marking sites for program, rearrangements of genetic material, and obviously I can go on and on, controlling RNA processing, editing, splicing, modulates translation process. Obviously, it even regulates embryological development in some sequences. All of this is found in the non-protein coding region, and the fact that we're finding more and more.
All right, so I'd like to ask you right now, do you even understand what my point was?
No, I understand what is the things that I'm listing. You're saying that it's in those regions, but based on that spurious transcription, it doesn't mean that it's...
No, that's not what my point is.
You're agreeing with the functions that I'm
Let me
saying.
put it this way.
You're saying it's such a small percentage of it that it means nothing.
Let me put it this way. Let's say you have the entire alphabet of letters, okay?
Yeah.
And I tell you that the letter A has a function. Okay?
Okay. Yeah.
Does E have a function?
So you're saying the letter
A
has a...
You don't know if E has a function.
We don't know. I admit that
it's in the same. It's in the same category. It's in the same category as the thing you know has a function, but you don't know that it has a function yet, right?
Are you familiar? Are you familiar with the article called "Immortality of Television Sets"? Because it was based on evolutionists who actually criticized ENCODE for not using evolution as a starting point. So it seems like there's this huge bias based on everything that has been discovered, because it's showing that these sequences aren't idle. Okay, so obviously a lot of just wait. So if a lot of this genome is transcribed, okay, and if it is just spurious transcription, I'm just saying a lot of energy for the cell to do something like that that doesn't actually have function. That's
so now let's move to another point, and
okay,
it's related. It's a very closely related point. Imagine, if you will, that you create a 10 kilobase sequence that is completely random, and you take that and you insert it into some simple eukaryotic cells, into some yeast cells, right?
yeah.
And then you get it incorporated into the genome, and you grow up these yeast, so you get a bunch of them, and then you do deep transcriptome sequencing on these things. So you have this big sequence, 10 kilobase, right, of completely random order. Okay. Now, if you were to find that of that 10 kilobase, that 60% was transcribed, it had at least one read from at least one cell in the entire experiment, that 60% of that completely random sequence was transcribed. And then you saw that from the rest of the genome of the yeast, that 80% is transcribed. What does that tell you? Because it should tell you something very specific.
No, it actually would tell me that, based on the fact that we know very little about the genome and we're discovering more and more every single day, that this does suggest and is a strong trajectory for genome-wide function. Because all I'm seeing from you is evolutionary bias.
Okay,
I'm also
so you've
seeing a
missed
rescue
the
device,
obvious.
and I'm
You have
seeing
missed the extremely obvious statistical implication there, which is that 60%
I know what
is noise.
I know it. I know that noise is the rescue device, but if you actually look
It's
at
not
one
a
of the
rescue
first,
device.
just
It
what,
is
I just want to say,
demonstrable. You
if you
can
look at one of the
demonstrate
first,
it.
if you look at one of the first junk sequences that we actually found doing something, it had to do with, and you're probably more than aware of this, had to do with males and females, because we know males have one X chromosome, females have two X chromosomes. Now, a double dose.
Yeah, you're talking about the thing I was just talking about.
Yeah, and what I'm saying is they discovered that because half the genes on the X chromosome are activated, while the other half will deactivate. So on the woman, so that actual important
It's not half the
function,
genes, half the chromosomes. It's
Well,
bar body formation. It's random X and activation.
well, it's half the genes of the X chromosome will deactivate, and the other half.
That's
wrong.
That's wrong.
No,
I'm sorry, that's
right.
wrong.
Let me explain myself without you actually interrupting. Okay, so what they found, because we know a double dose of the genes on the X chromosome would be bad. Okay, now half of the genes on the X chromosomes are deactivated, and the other half will deactivate on the other one. So anything a female needs.
Okay, so you want me to tell you why what you're saying is wrong?
Chromosome.
Okay.
How is that wrong?
You know, because what's happening is that one of the sister chromatids, right, is inactivated the entire length of it, okay, all the way up and down. It gets inactivated, right? So you have, right, one copy of each gene is inactivated, but it's always the copy from the same side. Okay, that means that that in this cell, for this chromosome, either the one you got that in this cell for the X chromosome, either the copy of X that you got from your mother or the copy of X that you got from your father is entirely inactivated, right? One or the other.
I'm talking about
up and down.
I'm talking about the woman, though. A double dose of an X chromosome is, I'm talking about the actual function that they found in these, because I think you're missing the point. What they found out is that a portion of the junk sequences was actually calling all the shots and was coordinating these activities that we're talking about.
Yes,
so all the
XIST.
Yeah, so all the junk sequence, obviously these sequences that aren't coding for proteins, actually tell all the other.
It tells you that one,
how to do it, how much to do it.
No,
I mean,
sorry.
so many overlapping roles there.
I'm sorry, you cannot make that assumption on the basis of that evidence. You can say from that it is possible. It is possible that a non-coding RNA can have a function that was already known prior to the discovery of the function of XIST. They already knew that. And so guess what? They were kind of looking for something like that because they knew those things could be functional. But the fact that things can be functional doesn't mean that everything of that same description is functional.
Exactly why I say the word trajectory every time. Let's just say, for now, for sake of argument, which I don't agree, let's just say it is 80% junk or whatever number you want to throw on it. Can you give me a step-by-step in the DNA on a genomic level of the very, very busy, I'm sure we'd agree with that, embryonic developmental stage? Not the growth factor, but what the DNA actually does step by step, building the embryo, aka all the construction tools, everything that goes into that. Can you give me that?
Can I give you that like right now in this hangout? No.
Now, a lot of that is unknown. And what we're saying and predicting is that a lot of that process is taking place in those non-protein coding regions. A lot of those developmental stages.
I encourage you to demonstrate that claim. It is not incumbent on me to demonstrate that claim of yours.
How I'm demonstrating is based on the 80% of the non-coding DNA that's transcribed into RNA that's suggesting function, based on everything that I've showed here. That's like what it is. It's a trajectory for this genome-wide functionality because we know very little about these embryo.
It's absolutely not
stages in the embryo. Because I urge you to go look it up. They know very, very little about that step-by-step process.
I encourage you to go look it up because they actually do know a lot about it.
It's simple, Crispr. We don't know what DNA does in development per se, and based on the fact that there's a strong trajectory here, we're saying, based on pre-existing heterozygosity, that it makes testable and falsifiable predictions. This is the prediction based on.
Why haven't you tested it?
Well, the thing is, test it. Okay, scientists perform knockout experiments to determine the function of DNA, obviously in non-human species. We know how that's done. In this way, obviously, the functions of sections of DNA can be determined, but comprehensive, we agreed to this last time, knockout experiments for a mammal have yet to be performed. So it is
just wrong about that.
So I'm saying it is premature for us to say that it's fully functional, but it's also premature for you to make the claim that the majority of non-coding DNA is non-functional. Are you saying that you've done these gene knockout tests on humans?
I haven't done them on humans, no.
And that's the point. So we need to know what's going on in the DNA on a genomic level during the developmental stages.
They've done them on mice, though.
But the thing is, have they done?
You understand that they have done large-scale genomic screens on mice where you would end up with 2 to 3 to 4 to 20 or however much megabase knocked out of a chromosome. Okay, and these mice, they'd keep breeding, they'd keep living. Maybe you'd see some minorly altered phenotype here or there from 20 megabase.
Yeah, but all we know is the growth factor of embryonic development. So, and that goes for mice as well.
Obviously,
they haven't looked at that in mice. So those construction tools have yet to be discovered in mice as well.
Now? Look, maybe you didn't hear what I just said. What I just said is that mice with huge sections of their genome knocked out have baby mice that develop through all of the embryonic development, also with the huge stretch of the genome knocked out, and they're fine.
Now, remember when I said a lot of these gene sequences that are involved in the embryonic development are silenced afterwards? If that knocked out region has been turned off after the mice embryonic stages, then that is very telling and very much expected.
Sorry. I think you still didn't hear what I said. What I said was you have, let me go into more detail. you have a mama mouse, right, and a dada mouse, and both of them have the same big ass region of the genome knocked out, and
Yes.
they have a little baby mouse, and guess what? Both copies of the DNA it gets from the mom and the dad are missing this big old section of genome. So it's never there in the first place in the fertilized egg.
How
So
do
it
you,
can't be present for any of embryonic development because it was never there in the first place. So
which
you can't
section,
activate it.
yeah, how much of this section of the genome that was knocked out, what makes you say that that is that specific section that is involved in the embryonic development?
Well, let's see. If they do this repeatedly, which they have, and they knock out lots of different sections, which they have, they can cover a lot of the genome. And guess what? Most of it doesn't do much.
So would you say that you look at a lot of this in an unbiased point of view, or would you say that you look at it in more of an evolutionary bias point of view? Like, do you suspect any type of future discoveries? Like, would you put that in print and make that prediction that what we're going to find uh, is going to be just more non-functional coding regions in these areas, or do you suspect that we're going to find more various forms of functions? And secondly, in regards to the mice, how much percent of the mice did they actually knock out? Because I want to hear the numbers, and obviously not just stories, in order that I can accurately address what you're saying. Go ahead.
All right. So, firstly, let me see. Yeah, here's the one I want. Yeah, I think this is the one. Ian, these are the ENUs I want, I think. Early methods, sure. ENU. Yeah. In ethyl in nitrous urea, which used to create rats with by mutating somatic cells and all this other stuff. Yeah, so this thing would produce pretty big changes. I'm trying to see if I can find a big meta-analysis. I'd have to get back to you with the meta-analysis of it, because I know I've got some. I've got.
Okay, no, hey, that's fine. You can get back to me. But what I want to say is, if we actually look at the history of genetics, okay, and I'm sure you know about it, very well. Even back in the 40s, what was it known? One gene, one protein, one function. That means many things have been overturned. RNA, you can even look in textbooks. A lot of professors and scientists back then used to say that it's very much underappreciated. Okay, so a lot of this junk DNA is what I'm saying is decades ago. Biology, because you can see massive literature now showing unexpected functions of RNA, right? So we're looking at a highly compressed DNA code beyond the best computer code, because you'd agree, we write books, but we don't write books that can read forward and backwards. Even if you look at something like protein moonlighting, right? What did they see or what did they think of in the 40s? One gene, one protein, one function. Protein moonlighting suggests that these proteins are doing numerous things you wouldn't even anticipate. It's the main tool, or one of the main tools, I should say, for building the body, and yet it's a multifunctional tool. So what I'm saying is, the cell uses possibly, these redundant elements to slow and speed the process. I mean, based on this highly compressed DNA code, I would say that the trajectory is strong for genome-wide function, but obviously, we'll have to see what the future makes out for that, and obviously agree to disagree on that. But that's where we go to pre-existing heterozygosity, which you seem to have had an issue with before, based on the 4 alleles. Can you respond to what I said and then also elaborate on that?
Honestly, I'm still looking for the size of the average size of the knockout from these ENU experiments, because it's bothering me that I don't actually have that in memory, because I really should know that number.
Well, I think a lot of it too based, because obviously, you know, the whole point of us getting into a lot of this junk DNA discussion, you know, would be, are mutations neutral or are they near neutral? So it's like, if we look at these tiny mutations that I'm saying are,
talk about that, that's fine.
I'm saying if they're near neutral, and let's say an animal has a harmful near neutral mutation, okay? Obviously, it's not going to die out because
Right,
that mutation is not big enough to be noticeable. So now that animal, I just want you to follow me with this story. It's more of a story, but, or a hypothetical. That animal now lives on to reproduce, say with its offspring also acquiring that mutation. Obviously, if it affected the reproductive cells, but now those offspring not only have the mutation it got from its parents, but, and we said this in our last discussion, 100 to 300 entirely new near neutral mutations.
140 approximately in humans.
So let's say that. So that means these near neutral mutations are going to spread throughout the entire population. And when enough time has, say, gone by or elapsed, they will accumulate to the point where the whole population dies out. And that's what I mean by genetic entropy. It all comes back to whether these mutations are neutral, near neutral. Go ahead. What do you think about that hypothetical?
Yeah, that's not even theoretically possible. I mean, it's not even possible in theory.
If you don't think that genetic entropy is possible or a problem, how come you guys always have to invoke natural selection, and especially natural selection stronger in the past? So you're admitting if there's no natural selection or if it's not working effectively, then this problem known as genetic devolution is an issue, though, right?
So firstly, if you're saying that even if there's no natural selection, even if you had a like single-celled organisms that were all immortal, right? And nothing, you know, they had no death from natural causes. They had no predators, and they had infinite food resources, right? There'd still be selective pressure in this case for whichever one breeds the fastest. Okay, so you can't, on a theoretical level, not have natural selection. Natural selection is a necessity. It is.
I agree. No, I totally,
literally impossible.
I totally agree with that.
Okay,
I'm just saying, if you don't believe genetic entropy, I'm just saying, if you don't believe genetic entropy is a problem, then why are you always invoking natural selection as the rescue device?
Okay, so you can't say that we're always invoking natural selection if you also agree that natural selection is a absolute necessity in the system. The fact that it must exist within the system means it must be considered for all relevant topics, right? You can't ignore this thing that must exist in the system. That's like saying there's got to be wind in a system, but we can just ignore it when we're trying, you know, to I don't know, like shoot a rifle, right? We're gonna, you know, have long range shooting. Well, there's got to be wind, right? Even if there's no wind, you still have to consider wind, right? Because if you don't, it's not going to work so well, is it? Right?
So I understand what you're saying, but I'm just saying that it seems that you think as long as natural selection occurs, which I agree, it occurs, it's very important, as well as beneficial. I just want to finish this, as well as beneficial mutations. The problem, though, that it doesn't seem like you're understanding is that I agree a population can be undergoing this genetic decline due to the vast numbers of slightly deleterious mutations, even while this natural selection may be amplifying a handful of, say, beneficial mutations. But even though a species can undergo these minor adaptive, fine-tuning situations to its environment, it's still degenerating in many other ways. It's just like a car. I mean, you can change the windshield wipers, change the brakes, change the tires. Doesn't mean the car stopped degenerating. It will degenerate over time, just like our genomes, personally and population-wise. Go ahead.
Right. So you've just made a claim with no evidence, and I don't think you even realized you did it. Anyway, so if natural selection must exist within the system, and if therefore it must be considered when discussing the system, then talking about why it is that natural selection necessarily means that genetic entropy will not happen isn't invoking natural selection as a rescue device. It's doing what you have to do, because if you don't consider natural selection in a system that must have it, you're an idiot, right? Like, you have to consider natural selection in a system that has natural selection. How could you not? And if a necessary consequence of this thing that necessarily exists is that there's no genetic entropy, guess what that means? It means Sanford's wrong. Now, if you want me to explain why natural selection necessarily means that genetic entropy doesn't happen, I can do that.
Well, no, because I'm saying that, and I'm sure you understand, and maybe you're misrepresenting. I understand natural selection is important, right? Because adaptation or natural selection, it's going to explain fine tuning to an environment, but it's not exactly going to explain how these near neutral mutations can be removed, because obviously a neutral mutation is not recognized by natural selection because it's neither beneficial nor detrimental. And I want you to answer that and explain what you're about to, but I want to also address what you said about the evidence, because if we actually look at, say, Lenski, he's actually revealing clear evidence of genetic degeneration, because in this artificial environment and based on these adaptations, they're actually being accomplished by means of either a loss of function or even a loss of regulation mutation. So hence, obviously, a loss of information. So that means these E. coli, which he's done many generations of, so this would be a good experiment to look at, it involves not these functions that are actually not even relevant to the static artificial environment of Lenski's experiment, which means that these bacteria are actually getting lazy. So if you actually look at that over hundreds of years, Lenski's evolving, and I say that very loosely, strains of bacteria must logically go extinct. Obviously, if he maintained the artificial environment that long, but that's clear proof right there, because nearly neutral deleterious mutations will inevitably cause this bacterial genome to rust out, and we're actually seeing that now. So we're actually seeing the opposite of evolution, which he wanted to show. So that's a real-time example of it happening.
Actually, we're seeing the exact opposite of that, and I'll tell you why. It's because those mutations that you're seeing are beneficial in the context of the bacteria that are growing there.
Look in the side.
No, because these evolving strains are degenerating toward the point where they can only grow in a specific, extremely limited, and entirely artificial environment.
Yeah, but they grow really well there. So that is not a harmful thing for those bacteria.
No, because these strains are becoming increasingly disabled and will be increasingly unfit to survive in the real world. So
who cares? They're not in the real world. They are in the environment they're in. All that matters is whether or not it's beneficial in the environment you're in. Saying that it's like saying that, well, okay, so this mutation that makes you bigger and stronger is really a bad thing, because it'd be harmful in an environment where you needed to be smaller and weaker. What?
No, because
nonsense.
It's not nonsense at all, because the fact that they're becoming increasingly disabled, and even while a handful of loss of function mutations have been demonstrated, I didn't say I disagreed with that, say to increase adaptation to this environment, they're greatly outnumbered by other mutations that have been accumulating relentlessly apart from selection with no visible effect. So I've given you a clear example of adaptive degeneration, or you can even call it reductive evolution. It's a form of genetic entropy. You can deny it all you want, but there's your real life, real time.
I certainly misheard you, and I apologize for that, because you said that they were observing a bunch of mutations that were causing them to no longer to no longer be able to survive in other environments, which would be, I think, which would mean that they would be more adapted to the environment they're in, which is a good thing. But if you're actually saying that there's just completely neutral mutations that are accumulating, then that's not making them more degenerate. That's irrelevant, because they're completely neutral.
Okay, so can you make predictions then, say, based on your rescue device right now of natural selection? Because how strong does natural selection have to be in the past to, say, account for what we observe and know about these near neutral deleterious accumulation of mutations today? Because you're invoking natural selection. So how fast did it have to be in the past? And are you willing to put that on paper and make that prediction? Go ahead.
I don't know what you mean by how fast would it have to be.
Well, how many of these near neutral mutations would natural selection have had to act upon and actually eliminate in the population of these species? Because if that's the rescue device, because I've already said that I agree.
Well, the near neutral mutations would be eliminated from a population to the extent that they are actually relevant to the survival or fitness of the population within that environment. And it will, so in other words, if they are barely nearly neutral, they will be eliminated quickly.
So are there numbers, like how fast, how strong?
I don't.
What are the numbers for this?
Yeah, so if you actually go into ecology, you can find the mathematical explanation of how drift interacts with selective pressure, which is the thing that you really want to find. However, I am not an ecologist, and so while I had ecology a few years ago, I don't remember the numbers off the top of my head.
Which is
However,
fine.
the conclusion of all of this is it didn't need to be any stronger at any other point in time than it is now. It's all fine.
It just sounds like a bait and switch. It sounds like a prediction cannot be made, and sure, I'll go look into it. I doubt that there is one, which would mean that it is a rescue device based on non-science.
I'll give you a prediction. You want a prediction? I'll give you a prediction.
Give me a prediction.
It would be exactly the same as what it is now.
So exactly the same.
Give or take a few percent.
Okay.
Give or take a few percent.
Okay, so let's look. So we see that an enormous 100 to 300 new mutations occurring in every person in every generation. So that means if your parents have no mutations, you have 100 to 300. Your children will have 200 to 600, and your grandchildren 300 to 900, and so on. So that means essentially all these mutations are damaging us in some way. I mean,
No,
look,
that is a claim.
know they cause aging, diseases, cancer. No, you don't believe that most of these mutations are well, for one, most of them are unselectable if they're neutral. If we agree to that, that means they're invisible to natural selection. So I don't even see your rescue devices solving the problem.
If they're invisible to natural selection because they are actually neutral, then they are actually neutral, which means they're not harmful because they're neutral.
No, because it's like single rust molecules are invisible to a car inspector, but over time it will spread in the population.
Okay, then now at that point, you're making a claim that there is an actual deleterious effect to each and every mutation. That is a positive claim. Please provide your positive evidence in support of this claim. Because you can't, and I already know you can't, because it's demonstrably false. But please go ahead and do it anyway, because you've made the claim.
Well, for one, I told you just based on the fact that no nucleotide by its very existence can be neutral, because we know they take up space.
Right, and I explained in detail why you were wrong about that.
No. I'm sure you gave an explanation. You know what? Even a student in school, they're going to provide an answer. It doesn't always mean that it's the right answer. The fact is, these unselectable mutations are the Trojan horse of pond scum to people in evolution. They come in silently. We know they're being so small that they can infiltrate the entire population, and they can't be removed by natural selection. So the most important part is it is consistent with our model, and you don't have to agree with it. I've already given you clear evidence from Lenski's experiment, which you want to deny. Those E. coli bacteria, they're disabled, they're weak, they're lazy, they're on their way to genetic degeneration. Okay, so natural selection is not a god, it's not a deity, it's not going to solve all your problems. Go ahead.
Just to repeat for the audience, you're saying that you can't actually support with evidence any of the claims that you've made because all of your claims are actually demonstrably wrong. I'm fine with that if that's your position, but I don't think it helps you any.
No, I actually just supported the claim with a real-time example based on Lenski's experiment.
Right.
I also find it shocking that you don't agree that most mutations are deleterious.
I don't agree that most mutations are deleterious. I think there's no evidence for that.
You don't believe that there's so would you 99% of mutations you wouldn't agree are detrimental in a way to an organism?
I don't see the evidence to support that, no.
So you can actually even see with RNA viruses, I think it was the H1N1 when it entered the human population, I think it was 100 years ago. It did cause the death of 50 to 100 million people. These viral samples were collected over the decades, and those genomes were actually sequenced during the last 10 years. When they reconstructed it, they noticed that the original strain to all the subsequent genotypes of the next strains, they were actually degenerated, and there was a declining fitness. So it actually led to the eventual strain disappearance. I think it was in 2009, after more than 10% of the viral genome had mutated. So those are another example of a real-time, I guess, evidence of genetic entropy. So there's two evidence. I find it shocking.
So let me respond to that.
Go ahead.
So the genome size of that is 13.5 kilobase, and it has essentially 0 slack. That is extremely and completely different, and more or less entirely non-comparable to what we're actually talking about here, which is mammalian differences, specifically humans, but also all vertebrates, all animals, all plants, all eukaryotes, and most prokaryotes, and some viruses as well, because some viruses actually have very large genomes. It's not common, but some of them do. So yeah, basically you're comparing apples to oranges here.
Well, I would disagree.
I'm sorry. Anytime somebody says, "Oh, you want to know why that thing won't work in people?" Well, let me tell you about an example from a virus. I really have to wonder about how much understanding they actually have of biology.
Which is fine. You can have the audience go look up those examples and they can decide for themselves. But how come there's a huge increase in genetic related diseases if this genetic entropy isn't actually true? Like, have you looked at the database of actual, even based on pure point mutations? I mean,
because we started working on making sure people who had these things don't die right away.
But the actual list of these, I mean, there's so many diseases, whether it's autoimmune diseases, whether it's point mutation related diseases, gene duplication related diseases. I mean, they're just on the rise, they're increasing, and a lot of them have no diagnoses, they have no treatment, and they have no, or I should say, much research backing the pathophysiology of these.
So you're saying that there's no evidence that they're genetically related at all?
I'm saying that, well, I think we can agree that mutations such as point mutations, gene duplications, they do and are the result of many. Are you disagreeing that we're seeing a rise of genetic related diseases?
Okay, look.
I'm saying I don't know that it's evidenced by what you've just said. No.
Well, obviously, if we're accumulating, Athena, go ahead. What were you going to say, brother?
I can't yell. If you look at the side, Nestle posted something about that he debunks genetic entropy and whatever. I don't know if you're paying attention to the chat, Crispr, but if you look at the link I just posted, if you can share that, I'll present it and you can go through it.
Well, I want to get into.
Yeah, I never actually read this post by Nestle. I'm sure it's quite good though. He's very thorough.
Yeah.
I want to get into, and I'm sure we can do that, but if we look at all the DNA differences, CRISPR, would you say that those DNA differences, let's say nuclear DNA differences, are they the result of mutation, would you say?
Differences between what and what? Like
mutations, right? So if we're looking at the genome sequencing of, say a dog and a human, obviously we're going to notice especially genetic markers that are
your answer is between dogs and humans. That's a sufficient answer. You asked me what the cause was, if it was mutations or something else, and then when I ask you differences between what and what, you said, well, mutations.
I'm just is that what you assumed the majority of differences are
okay, so we've covered my first question. So when you say difference, do you mean difference in terms of centenary, in terms of, or just pure sequence? Like if you find
I would say if we're lining up the genetic sequences and we're looking at ATCG, ATs,
yeah, I
whatever
know,
it
but
is,
what I'm
every
getting
time
to
it's a
is what you
difference.
mean by lining up. Because if I have a sequence, right, and
Yeah.
in one organism, right, it goes from A directly into B, right,
Yeah.
and then somewhere else is section C, and then in another organism, A is off on its own, and B goes directly into C, but within A and B and C, the sequence, nucleotide by nucleotide, is exactly the same between the two organisms, right? How different would you say they are? Is that one mutation moving B from being next to A to being next to C? Or is it however many nucleotides are in B worth of mutations?
I would say, however, I'd have to actually look at an actual, I'd have to see a screen share of this. But yeah, I'd say based on the single nucleotide differences between whatever, two or however many.
Well, I'll tell you that in genetics analysis, that would be one mutation.
Okay,
That
so
would count as one difference.
so that one difference would be attributed to a mutation, then, is what you're saying.
Yeah.
Okay, like, are you familiar with Mendel and his pea plant experiment? I know me and Immutable Destiny talked about this for a little while, but
Yes, I'm reasonably familiar with Mendel.
like, so when he was looking at traits, he realized that they remained distinct, as in they didn't blend. So they appeared and disappeared in family trees. So in other words, obviously they came in dominant and recessive types, right?
Yeah, Mendel was sort of lucky when he was doing this, that he was working with pea plants, because they have a really unusual abundance of of traits that have very high penetrance and expressivity. And so if he had been working with dogs, for instance, I mean, ignore the numbers problem, right? Even if he'd been working with fruit flies, he would have had a much harder time. Pea plants are just, they assort really, really cleanly, and that's actually very uncommon in most life. So most of the conclusions that you might try to draw from it are actually very, very shaded by this. That you're trying to apply assumptions that are based on a minority case to the majority of cases, and that's not really a good way to go.
Standing before you respond to that, Nestle has a super hard on about somebody reading
Why
from that
doesn't he
post.
come in?
Okay, I'll see if he wants to come in. Hold on.
Well, just wait a minute. So yeah, you can bring him in. I don't care. But I just want to say that obviously when it comes to these, I think the main point to take from Mendel would be what he noticed in regards to dominance. And obviously, because Mendel assumed pre-existing differences and a functional sequence of heredity. So if I actually look at, what we reject, we obviously reject the assumption for nuclear DNA. That explains the vast majority of DNA differences as mutations, right?
You can't come
We
in.
would say that these differences existed in a heterozygous state in the ancestors.
Yeah,
So if I'm saying that most nuclear DNA differences are the result of created heterozygosity, that would be a huge claim and then a huge, obviously, indication on how we could get the rapid speciation that we see and know today in living organisms. Go ahead.
Yeah, so the problem with that is the concept of haplotypes, which basically completely destroys your entire argument. Because if you have only two organisms, which is what you would have following the arc, right? For not for humans and not for the clean animals, but whatever. The majority of cases, you have two. So even if you assume for the moment that each pair has for each gene a total of 4 different alleles, most of these alleles will actually not independently assort. And this is the thing I was trying to get to with the pea plants, because chromosomes are single packets, but they have recombination. But the likelihood of recombination is dependent on distance, which means that if you have two genes that are very close to one another, you will not have an equal likelihood of getting any possible combinations. You will almost certainly always get the same combination. Sometimes not, though. But where this becomes relevant is that it will become virtually impossible if you have so let's just look at two genes, right? You have A1, A2 in the father for the first gene, and B1, B2 for the second. And then in the mother, you have A3, A4, B3, B4, right? Now, getting a organism that has A1, A3, it's virtually impossible if these two genes are right next to each other. Will not happen.
Okay, first I want to go back to, okay, and I want to comment on everything, but let's start with, because if we're actually looking at 4 alleles, okay, I agree that that means we cannot get much diversity, but if we actually expect low genetic diversity based on this, and the fact is that's what we actually find, that means that 4 alleles wouldn't be a problem as a starting point, say for 2 original humans in regards to Adam and Eve. So that would actually be a better prediction based on that low diversity from those 4 alleles pertaining to what we see today.
Yes, except that when you talk about the diversity that we see today, you're thinking about it in terms of phenotypic diversity. However, we see an enormous variety of genotype diversity without phenotypic distinction, which means that you have to be able to explain why it is that you have 2 people who have 2 different mutations right next to one another, right? So in other words, person A has a mutation on base 1, and person B has a mutation on base 2, and they're completely different from one another, right? Single nucleotide polymorphism. You have a very difficult time explaining that. Or, more to the point, single nucleotide, who cares, right? How about an ERV? How about an STR? A STR would be very difficult to explain, especially some of them that are extremely rare variants. Doing that would be a great difficulty. And then you have the fact that if you look at STRs, you don't just have 1 or 2 or 3 or 4 alleles. For some STRs, you might have several dozen different alleles.
Okay,
let me respond. The approximate rate at which the new alleles occur, and that rate of formation just does not fit your timeline.
Okay, let me respond. You can tell me if I missed out on anything you said. In regards to obviously Adam and Eve, and then obviously the diversity we see among humans, it wouldn't be about phenotypes because 99.5% of us are identical, whether it's me, you, whether it's someone in Africa. That means we are all genetically similar in regards to that, and it would fit with low allele starting points. And if we actually define, say, a single genomic or an allele as a single genomic position, which would be independent of its relationship to a gene, that means enormous allelic diversity can be generated by mutation. But as an aside, allelic diversity, I said this before, and I think you kind of directed towards this, it need not arise by mutation. Because if I'm if the genomic position definition of an allele rather than say the gene unit definition, which I think you were using, other mechanisms besides mutations can generate allelic diversity. For example, a single gene typically spans thousands of nucleotides, which means, and say single nucleotide variants might be distributed throughout the gene. For example, and I'm almost done here, 90 of the nucleotides within the gene, if we allow for the genomic position definition of alleles, every single one of these 90 single nucleotide variants that I'm talking about may have existed in a heterozygous state in each of these individuals of the pairs brought on board the ark. So now I perfectly explained Adam and Eve and the human diversity, as well as based on
so the problem though is that each of these variants cannot independently assort. That's the thing you can't explain.
So what you're saying, if we start off with heterozygous organisms, visible distinctiveness, it's not going to be quick based on recombination and gene conversion, which I'm saying would provide many new varieties of combinations of traits quickly.
Yes,
because
that is actually the case. Yes,
but
if you only have two organisms, if you only have two organisms, you will not get extensive variety from them.
But are these differences, are you assuming that they're built in, like, front-loaded differences, not based on mutation, based on functional created differences?
It absolutely doesn't matter. It doesn't matter. It's like asking me, well, if you add 1 plus 1, are you going to get something other than 2? Well, what if we front load the 1? What? No, it doesn't matter. That doesn't even make any sense. That's just the math.
No, because what I'm like, for example, if evolution started with thousands of people, like the out of Africa scenario, then our genetic diversity shouldn't be low. So what I'm saying, at least in regards to Adam and Eve,
that's just a misunderstanding of population genetics, though.
Well, if it's not about, because their population bottleneck was due to low genetic diversity, how come the evolutionists then? How come you're copying our flood population bottleneck, given the evidence that we see?
Well, I mean, we're not copying it.
Well, we see genetic diversity based on the 4 alleles you even adverted to. So if 4 alleles is no problem as a starting point for 2 original humans, Adam and Eve, that's what we see. 99% identical. This fits with low allele starting points, right, low genetic diversity. I mean, I don't see a problem at all.
So firstly, like, you understand that you're talking about like 2.7 million mutations per year. If you say 99.5% similar and 6,000 years, that's like 2.7 million mutations per year, which is going to be a real trick.
Well, the allelic diversity, it's not generated strictly by mutation. I've already iterated that a few times, especially based on those pre-existing DNA differences.
Because of haplotype. Has to be because of haplotype inheritance, because they don't independently assort. So most of these variants will be lost in the first generation because they do not independently assort. This is the thing you do not seem to understand.
Are you saying there's too many mutations that we're seeing in order for what I'm saying about the population bottleneck?
Yes, that is basically right. Yes.
But how come evolutionists seem to underpredict the nuclear DNA differences, but overpredict the mitochondrial DNA differences? Because it still seems to me that you're assuming mutations as a starting point, because the DNA differences and absolute DNA differences. If you are assuming what you're saying in regards to all mutations as compared to functional created front-loaded differences, I don't see where there's a problem. Because if my bottle defines, I want you to comment this, if I'm defining alleles in terms of DNA position and not just individual genes, the maximum of 4 versions of each gene could be present in the, it's going to allow for that speciation. And even after the flood, you'd see that radiation and after effects of the flood. You probably would see the sparking of incredible.
Okay, so the problem is, and I will explain this once more. I will try to use smaller words. If you define it by position, then that means that as you're understanding it, and your understanding is wrong, and I'm going to explain why, then what you would have is literally every single possible combination of a genome that is that long, which would include, by the way, for humans, the mouse genome. And obviously that's wrong, because the mouse genome is smaller than the human genome. It would also include all viral genomes, all bacterial genomes, all other genomes that are smaller than 3.2 gigabase, if that's the way you're doing it. But the problem is that you're ignoring haplotype inheritance, because you still do not seem to understand that the genome, contrary to what Mendel said, does not independently assort at the level of the gene.
Well, for one, with Mendel, 1 to several new traits per generation, it has been revealed. That means the speciation of 4,000 years
Can
would
you
not be
please
a
repeat
problem.
back to me what I just said?
It still seems to me that you have a false assumption.
No, I'm
and
sorry.
a false
I'm sorry.
idea of what our
This is
starting part is.
extremely important. Do you understand that the genome does not independently assort?
No, I don't see why what you're saying.
Do you understand that that is a statement of fact?
Okay, do you agree that millions of DNA differences separate species right now? I just agree with that.
Do you
Yes or no?
understand that the genome does not independently assort? I need you to understand this, because this is fundamental to why you are wrong.
But having an understanding of that does not prove.
It is going to go there. If
Hold
you
on.
understand it, it'll make sense. But
Hold
you
on.
don't understand it, and you seem to be really reticent to try and understand it. So let's do this again. Do you understand that the genome does not independently assort?
I want you to elaborate what you're saying, because I think you're trying to step on me with being a scientist, and I'm not going to let it happen. So why don't you actually explain?
Happy to.
I'm happy to. But I want you to, and I want you to explain it, but I want you to say how that's going to be a problem, because we know that millions of DNA differences do separate species, okay? And each generation, mutations generate a trivial amount of variety. So to generate millions of DNA different, in order to generate these millions, I just want to finish what I'm saying, and you can continue.
No, you asked him to elaborate, and he said he would.
Because your model requires that these differences be, as you say, front-loaded. However, for them to be front-loaded and explain the differences, they must, all of these differences, independently assort. But that does not happen.
So you're saying it doesn't, because it does sort independently.
It doesn't, though. Now, again, I'm trying to explain this to you because you haven't had genetics. Mendel was almost right about it, because genes are carried on chromosomes, and the chromosomes themselves independently assort. And also, you get recombination between chromosomes, which means that if you have two genes from the opposite end of the chromosome, there is an approximately 50-50 chance of which one you get. So they mimic the independent assortment seen for the entire chromosome. However, if you have two base pair right next to each other, the odds of you getting base pair 1 from chromatid A and base pair 2 from chromatid B approaches 0. It is almost 0. Now, as you move your nucleotide 1 further and further away. Let me finish.
I'm trying to follow you. Are you saying that they, because from my understanding, they shuffle, it's random, and they don't sort? Are you saying that there's like, are you saying that they're sorting?
I'm saying that, have you ever heard of centimorgans? The unit of measurement used in genetics, centimorgan?
Can you define centimorgan for me?
Sure. Yeah, that's what I was actually trying to do earlier. So basically, the maximum measurement in centimorgan that you can determine with a single experiment is 50, right? And what that represents is a 50-50 chance of two genes independently assorting, okay? That means that if it's lower than that, you have what's called a genetic linkage, which means that these two genes, if you know that you have gene 1, then you know that there is an increased likelihood that you will have gene 2. And what that tells you is that these two genes are located very close to one another in the genome. And the closer they're located to one another, the higher the likelihood that you get both of them. All right? Because they're not assorting independently. All right?
Okay. And keep going. I just want to know, what is the meaning of pre-existing differences when I say it?
Well,
like when I say pre-existing differences, what do I mean when I say that?
I assume that what you mean by that is that the originally created organisms have a greater degree of allelic diversity than what we see now.
So if we're looking at these, say, tens of millions of nuclear DNA differences that you're saying are based on mutations, what am I saying they're based on?
You're saying that they are based on the fact that they were already there.
In functionally front-loaded, created, pre-existing differences.
Now,
So
the problem with that is...
No, wait. So if the pre-existing differences then are already scrambled, the assortment you're referring to is already there. So
no, but
I still don't see how that becomes a problem.
I will tell you why. I will tell you why it's still a problem. The problem is that if you have allele 1 for gene 1 on chromosome or on chromatid 1 in Adam, and you have allele 1 for gene 2 on chromosome 1 or chromatid 1 in Adam, right? And they're right next to each other, then you will always get allele 1 for gene 1 with allele 1 for gene 2. You will never get a different variety than that specific inheritance. It doesn't matter that chromatid 2 for Adam has allele 2 for gene 1 next to allele 2 for gene 2. All that means is that if you have allele 2 for gene 2, you're going to have allele 2 for gene 1. You will not have allele 1 for gene 1 and allele 2 for gene 2. It will not happen that way.
I get what you're saying. You see it as a major problem. I don't see it as a problem based on the pre-existing differences already there. This is my answer. It's already there. Scrambled starting point. The more scrambling as time goes on will lead to much faster speciation and unlocking of pre-existing traits.
No,
I don't see it as a major problem. You can leave it for everyone else to decide.
One at a time,
the
guys.
haplotypes,
One
right?
time.
So when you have an offspring from this original couple, right? And this offspring only gets two sets, one from either parent, which means you lose the other one, the other pair. Now, maybe they have a bunch of kids, right? So there's each organism is going to have 20 kids. Okay. Now, well, they're going to have kids, right? But what you will never see, though, is allele 1 for gene 1 with allele 2 from gene 2. You will never see that. And yet we see a bunch of really weird combinations, and that tells us there must have been a lot of time here.
Well, that still doesn't explain why evolutionists would over-predict the mitochondrial DNA differences and under-predict the nuclear DNA differences. So it sounds to me like you're underestimating our pre-existing differences because you must only be thinking it's few, but it's actually quite a lot. So I don't understand why you don't understand it. And you know what? That's okay, because you not understanding it is not really my problem anyways. But it's been a good conversation. You guys can continue it on. We'll let everyone else decide. I really just feel that it's something you don't understand based on these pre-existing differences. You really are underestimating it and underestimating the entire model. But you would also have to make those testable, falsifiable predictions based on the mitochondrial DNA differences and nuclear DNA differences yourself. Because obviously evolution is under-predict it and over-predict it.
Yeah, Jay, go ahead.
Where did that burden come in? Where did how did you just throw that burden at CRISPR when you're the one how did you just do that?
Because science has to be testable and falsifiable. If we're looking at nuclear DNA differences,
conveniently unlike religion.
Yeah, I don't understand. You threw something at CRISPR. He seems to have had a little bit more he has a little bit more understanding of the science than you do. You have more understanding of the science that I do, but now you're throwing a huge burden on him because you want something to be the case, and he's telling you that's not the case.
No, the case is the nuclear DNA differences that I'm talking about, it's better explained based on pre-existing heterozygosity, because evolutionists under-predict the amount that we see, and they over-predict the amount of mitochondrial DNA differences. It's all the same thing.
It actually isn't better predicted by your model, because your model would be consistent with absolutely any possible description of events.
Okay, well, that can be elaborated on next time. It's been a good conversation. No animosity at all. I think it's been fun. It's been long. I need some water. Listen, guys, don't party too hard tonight. And hopefully we can do it again soon. Take it easy, boys.
Bye.
Bye.
Okay.
Okay.
What was the motivation there? What was he trying to go for?
I mean, you already know mine, but yeah, it's Crispr.
I think victory by fatigue was what he was going for.
Well, that's basically what I said, yeah.
Yeah, because that. Anyway, wow. I don't know.